Intratympanic delivery of aldosterone via a hyaluronic acid hydrogel by Carlos Cruz, Joshua Hachirian, Tomoko Stansel, Matthew Martin, and Carson Zide.
Our company, Hydro Audio, is a mainline manufacturing and R&D pharmaceutical company. Our company's objectives are to produce a therapeutic hydrogel that impregnates aldosterone in hyaluronic acid for diffusion-driven delivery into the cochlea for treatment of neural age-related hearing loss, and also to facilitate application of the therapy by designing and producing a custom injector that increase the safety and minimize the impactness of intratympanic delivery.
So, a little bit about the motivation of Hydro Audio into producing this product. So, approximately one third of Americans between the ages of 65 to 74, and further half of over all after the age of 75, are affected by age related hearing loss. Now, recent studies have shown that as you age, aldosterone levels decrease inside of the auditory system. Recent studies have also shown that aldosterone preserves the connection between the auditory system and the brain. This connection are essentially made up of nerve cells that are preserved when aldosterone is present. So Hydro Audio wants to deliver aldosterone into the inner ear, avoiding any kind of systemic delivery routes. Systemic delivery routes would lead to high blood pressure and potassium dysregulation, which would lead to a whole range of different diseases. There are two main barriers that aldosterone would have to cross, the blood labyrinth barrier and the blood-brain barrier, meaning a very, very high concentration of aldosterone would have to be administered, and then you would hope that some of it would reach the cochlea, if any. Now, there are no current treatments to treat age-related hearing loss. There are currently cochlear implants and hearing aids, which are intervention techniques, but these two treatments rely on the degenerating nerve cells that we are trying to preserve with aldosterone. Hydro Audio's approach towards designing such a proposed product and mass producing it was undertaken by a five-step methodology. The first step was figuring out correlations that could describe aldosterone's transport through the round window membrane and a hyaluronic acid hydrogel of set composition. Secondly, a governing equation was selected to describe mass transport as the basis for our model. Third, COMSOL in order to prove that aldosterone was diffusing through the round window membrane and into the cochlea. Fourth, computer-aided design to redesign a delivery method. Lastly, market analysis in order to prove that this proposed product and process would be feasible and profitable. Shown on this slide is a summary of our product design. To the bottom left, you can notice the eardrum represented as a white oval, a needle that protrudes this eardrum and delivers the hydrogel plus aldosterone, our product. To the far right, you can notice the round window membrane, a membrane composed of two outer epithelial layers and a middle fibrous cell layer. Now, to the top left, you can notice where this round window membrane is located. It is located in the middle ear, allowing a membrane connection into the inner ear. The inner ear would be the cochlea, shown as that snail-like structure. So a little bit more into how the product specifications for the hydrogel were selected. So one, we wanted to deliver 1 milliliter of drug containing 0.375 milligrams of aldosterone to each ear. 2. The 0.375 milligrams of aldosterone would be expected to diffuse across a 3-day span into the cochlea through the round window membrane, or 0.125 milligrams per day. Thirdly, the hydrogel was selected to be composed of 90% thiol-modified hyaluronic acid and 10% thiol-modified gelatin. Such a composition has been shown through previous literature to provide around 3 days of sustained delivery. Fourthly, transport is assumed to be all diffusion dependent and absolutely no convection. Fifth, aldosterone in the cochlea will be assumed to be approximately zero in order to enhance this diffusion model. Sixth, the aldosterone in the hydrogel will be assumed to change very little relative to the hydrogel being depleted through the round window membrane and into the cochlea. This will give us a quasi-steady state type of assumption that we could implement into the model. Now, moving forward, the following picture shown on this slide represents how our product is being administered and how it is working. So, as you can see in the picture, the blue is representative of our aldosterone hydrogel product. And as you can see, that is about 1 milliliter of the hydrogel aldosterone product containing 0.375 milligrams of aldosterone. Further, you can also notice that there is diffusion occurring into that round window membrane section, shown in the red circle. And to the left, it's being delivered by a hydro audio injector, shown by the arrow. Moving forward, how did we construct the round window membrane encompassing its actual physical geometry and characteristics? Well, experimental data and literature has provided all of that information to us. So the physical characteristics of the round window membrane are as shown. It encompasses a diameter of 1.8 millimeters, a thickness encompassing those 3 layers previously described of 70 micrometers, and, as previously described, a material of 3 layers, essentially cells, fibers, and cells. On the bottom left, you can see a representation of the round window membrane, and on the top left, you can see relative to where its location is in the auditory system, as shown previously as well. Okay, so now let's talk a little bit more into how we were able to model aldosterone diffusing through a hydrogel. So in order to do this, the simplest way was to acquire a diffusion effective. So essentially an effective diffusion coefficient that would incorporate the characteristics of the hyaluronic acid hydrogel at its set composition and would also incorporate aldosterone passing through it. Now, we were able to find a correlation. That correlation is a function of the diffusion coefficient of aldosterone transport in water, which is further a function of the molecular weight of aldosterone. Secondly, it's a function of the hydrodynamic interactions that aldosterone has with the hyaluronic acid hydrogel. And third, it's a function of the pathway tortuosity that aldosterone finds itself. In our case, the effective diffusion coefficient of aldosterone in our hyaluronic acid hydrogel at 90% hyaluronic acid and 10% gelatin resulted in a value of 4.2 times 10 to the negative 10 meters squared per second. So now let's talk about how we modeled aldosterone's diffusion through the round window membrane. So previously we spoke about finding an effective diffusion coefficient for aldosterone in the hydrogel, and now we set out to find a correlation that would describe an effective diffusion coefficient for aldosterone in the round window membrane. So that was accomplished by finding a specific correlation that would incorporate the molecular weight of aldosterone and model it passing through an epithelial cell system. Now, in our case, as can be shown from the equation, the round window membrane is of course composed of 3 layers, and through that physical characteristic, the A coefficient was found to have a value as shown on the bottom right as 1.778 times 10 to the negative 4 centimeters squared per second, and B was found to be a coefficient of 0.75. By incorporating these two values and knowing that we are dealing with aldosterone, which has a molecular weight of 360.44 grams per mole, we were able to find an effective diffusion coefficient of aldosterone through the round window membrane equal to 1.97 times 10 to the negative 10 meters squared per second. Okay, so the mathematical modeling involved for our system and for aldosterone's transport through a porous media transport was selected to be governed by Fick's second law that incorporated porosity and tortuosity. Now, as can be seen below in the first equation, porosity and tortuosity is incorporated in order to find the effective diffusion coefficient that was previously shown in the previous slide. Now, when porosity shown by that E and tortuosity shown by that tau is multiplied by the aldosterone diffusion coefficient, it provides this effective diffusion coefficient. Now that can be applied into our second equation, which is Fick's second law for non-steady state mass transport. And through that, we can begin to model the transport of aldosterone through the round window membrane and through the hydrogel, and then eventually into the cochlea to provide a therapy. Okay, so now let's look at some results. So all of these results were acquired through COMSOL modeling, and COMSOL has the unique feature of allowing us to look at the physical dimensions of a proposed product. So in our case, on the left side of this slide, you can notice that the blue is the round window membrane cochlear boundary layer, which has a concentration of approximately 0. All of the red is hydrogel. Sandwiched between this hydrogel and blue layer is the round window membrane. Now, to the right, you can begin to see that at time equals 0, there is no pronounced concentration gradient because the drug has just been administered to the middle ear. But once steady state begins to develop, you can begin to see a concentration gradient developing. Now, all the way to the bottom, we can see just the round window membrane, defined as that 70 micrometer thickness, that there is a concentration gradient occurring, and there is no longer a concentration of 0 beyond the round window membrane. This proves to us that aldosterone is passing through this round window membrane and being delivered as therapy into the cochlea. Now, more specifically, let's look at a 2D model of the previous representation showing at initial delivery time. Now, as you can see on this graph shown on the leftmost side of this slide, you can see that the red is once again presented as the bulk hydrogel containing 0.375 milligrams per milliliter of aldosterone. Then you can see the hydrogel round window membrane layer, where you can start to begin seeing that pronounced concentration gradient. And then the round window membrane. Now, at initial delivery, there's not much transport, so most of everything from the round window membrane to hydrogel round window membrane boundary layer is to the right, shown as blue, or as very low concentration to 0 concentration. Also, to the right side, we can see two pictures depicting the top lateral view and the bottom lateral view of this transport. Now, this is important because you can see that as aldosterone begins to diffuse into the cochlea or through the round window membrane, you can begin to see that at the very, very top and at the very, very bottom, there is surface friction present. And this model very much incorporates this physical phenomena and therefore enhances the model. Now to look further into what is occurring at steady state or just after initial delivery. Now to our left, we can once again see the bulk hydrogel layer, the hydrogel round window membrane layer, just around window membrane, and then the cochlea. Overall, we see a more pronounced concentration gradient that has developed across all layers. This is indicative of transport occurring and aldosterone being depleted from that bulk hydrogel and into the cochlea for therapy. Now on the rightmost side of this slide, you can once again see the top lateral view and the bottom lateral view. Once aldosterone has began to move, indicative of flux, it does encounter this surface friction on the topmost side and the bottommost side of these cells, and therefore it's almost, as you can see, a no-slip region as diffusion occurs. Now this is good because it could also prove to us that diffusion is occurring, and therefore overall our product is delivering aldosterone into the cochlea. Now overall, this provides us grand evidence that our product does deliver aldosterone through the round window membrane and into the cochlea to treat age-related hearing loss.
Our custom aldosterone injection pump is shown below. The delivery tool is called the hydroaudio injector. The elevated needle allows the doctor using the device to have a clear view of the eardrum during the procedure. The needle's diameter is 40% of the size of the round window membrane to reduce the invasiveness of the procedure. The pump will have a delivery trigger that will administer exactly 1 milliliter of aldosterone hydrogel. Shown below is our aldosterone reservoir. Each reservoir will be filled with 1 milliliter of aldosterone hydrogel, which will treat one ear. The reservoir is designed to be quickly and easily loaded into the injection pump. The reservoirs are made out of stainless steel in order to streamline temperature regulation at 4 degrees Celsius.
We need to make some key assumptions in order to design the process in which we will manufacture the hydroaudio injector and the hydrogel product. First, the raw materials will be purchased in bulk, and in particular, D-isomer aldosterone must be of pharmaceutical grade. We will contract 2 companies to manufacture some of the parts for the injector. One company will produce the injector case, the other company will make the reservoirs and some of the injector pump components. Assembly of the injector will take place in-house, and the reservoirs will be sterilized in an autoclave. We will use a batch process to polymerize the hydrogel. And here is what that batch process looks like. The thiol-modified gelatin and hyaluronic acid will be the monomer mixture, and the PEGDA will be the crossing linking agent. All of our organic components must be kept at negative 20 degrees Celsius to maintain their integrity. The monomers must also be sealed, as exposure to air will cause polymerization to occur. To start the process, the raw materials are brought to a room temperature. Degasified, deionized water is then added to the monomer mixture and fed to one heater, while aldosterone and phosphate buffered saline are added to the cross-linking agent and sent to another heater. Both solutions are brought to 37 degrees Celsius and then mixed in the polymerization reactor. The reactor effluent is then sent to a separator to remove any unreacted monomer and cross-linking agent from the hydrogel. The product is then washed and dried before filling the reservoirs. Now that we have an initial design for our process, we can analyze the profitability of our project over the course of 10 years and determine if we should proceed with the project as is or look for new alternatives. As an estimate for demand, we took 10% of the population to be potential customers, which is roughly 1/3 of the people who reported age-related hearing loss in 2017. We assumed that each patient getting one treatment for each ear on average would get 4 doses in a month. Because the hydroaudio injector is reusable, most of our profits will come from the hydrogel-filled reservoirs. Since there are no other companies manufacturing hydrogel treatments for age-related hearing loss, our company would have a monopoly on this type of product. Before we can perform a feasibility study on the project, we need to make a few assumptions. For typical pharmaceutical plants, tax rates are around 35%, operate for 10 years, and take 2 years to construct it. For this project, we will use MACRs to account for depreciation. The labor costs come from typical salaries and benefits for chemists, engineers, operators, sales representatives, lawyers, and so on. Research and development costs are usually around 16% of the total revenue, and the market interest rate is assumed to be 20%. By using discounted cash flows, we can graph how the net present value of the project will change over the course of the plant life. As the graph indicates, our base price has a payback period of only a year and a half. Our revenue will come from marking up our raw material costs and other expenses by 20%. Based on this analysis, the base price of $101.50 is likely to make this project very lucrative with a discounted cash flow rate of return of 76%. This table shows more details of the feasibility study. The total capital investment was found to be approximately $500 million, while the taxable income was nearly $380 million per year. Our biggest impact to the net present value is in year 2 at $500 million. This is likely due to the tax breaks for depreciation are the largest in that year, the inclusion of the tax breaks for startup costs, and the impact of time value is very small in that year. In the end, selling off the land and recovering the working capital had little effect in our final calculations. However, with a net present value of $890 million, this feasibility study confirms that this project is highly profitable.
Approvals for drugs. We will be selling hyaluronic acid hydrogel as an off-label drug. Off-label use is the use of pharmaceutical drugs for an unapproved indication, dosage, or route of administration. When a drug has already been FDA approved, generally the interest in running clinical trials and gaining new indications for the drug is low because the clinical trial cost is so high. Currently, 20% of generic drugs are prescribed for off-label use. In the future, we will move forward with full FDA approval and have set aside money in our budget for FDA approval. Safety for this process. Most of the raw material must be maintained at -20 degrees Celsius with a backup freezer to preserve them in the event the primary freezer fails. HydroAudio's control system will be calibrated to only contain the necessary dosage, with quality tests of each batch to ensure proper dosing. All reservoirs are sterilized at the plant before entering manufacturing processes. Gelation occurs in an anaerobic environment which allows for a safer process. Polymerization is an exothermic reaction and a constant temperature must be controlled to avoid loss of product and prevent a potential runaway reaction. To prevent this, the reactor has a cooling jacket with a fail-open valve controlling the supply of water. What-if hazard analysis. Shown here is our what-if analysis of the process flow diagram. What-if scenarios are a process hazard analysis tool to identify hazards or weak spots within a system. Questions are asked on how a certain scenario might create a hazard. It can be used for any activity or stage of design. As shown, many potential system failures are taken into consideration with safety and preventative measures and recommendations for each potential hazard. Environmental impact. Environmental impact was assessed using the WAR algorithm to predict and identify the likely environmental impacts of our project, including the detailed elaboration of alternatives. The main impact of our product is in the event of a leak causing runoff and groundwater contamination. Also, the dryer will have a slight impact on the ozone, although very minor due to the overall small amounts of product needed for treatment. Key decision variables. Decision variables are unknown and controllable parameters of the system which finding their value is the purpose. Some of our discrete variables include the hydrogel material, a batch or semi-batch process, and the patient demand. Our continuous decision variables include the concentration of aldosterone inside of the hydrogel, the pH of the polymerization solutions, price markups, and the reactor temperature.
To optimize the process, we decided to recycle non-reactive polymerization reactants and unimpregnated aldosterone from separated back into reactor. For product, we ensure optimum safe aldosterone transport is occurring throughout 3 days by using COMSOL simulation of diffusion through round window membrane. We also used Aspen Plus for parameter estimations involved in process design. There are 4 key uncertain variables we have to consider. First, product price. Second, permeability of the round window membrane due to hyaluronic presence. Third, product demands. And fourth, R&D reinvestment.
In conclusion, aldosterone is shown to successfully diffuse through the round window membrane and into the cochlea when delivered via an intratympanic hyaluronic acid hydrogel. Our custom HydroAudio injector reduces invasiveness of the procedure and improves patient outcomes. The process is feasible in producing the required demand in a pharmaceutical plant. The product of hyaluronic acid aldosterone hydrogel and HydroAudio custom components is a highly profitable endeavor with an NPW of approximately $890 million for 10 years of production. The majority of the profit will come from the injection because the HydroAudio system is reusable. For this project, our DCFRR is 76%, which is standard for the pharmaceutical industry. This further incentivizes the investment.
Slide 32. Over the course of this project, there are a few recommendations along the way we think would be prudent to implement. The first is to expand production to new treatments for different illnesses. Some potential treatments could apply to sudden sensorineural hearing loss and some types of vertigo and tinnitus. It is likely that the future projects will need to utilize multiple plants or expand the capacity of our current design, since it is predicted that the population above 65 years of age will double by the year 2050. Because we would have a monopoly on this product, it would be easier to keep the competition to a minimum if we were to obtain full FDA approval to sell under the HydroAudio brand. This would be the focus of the research and development department. We should also start the production of aldosterone on site, as there are not many manufacturers for this product.
We'd like to thank you to Dr. Snow and our TA Aaron for their time and continued feedback and overall guidance throughout the project. We also like to thank you to Dr. Frisina for his preliminary technical feedback regarding aldosterone function.